Hello, I'm Pohing. My group and I have put together a short video showing how our immune system deals with an influenza infection. Let's look at the main antagonist here, the influenza virus. It is an enveloped virus that contains 8 single-stranded RNA segments, which are important molecules in the detection of virus. There are many immune cells involved in the response mounted against the virus. We have our phagocytes, especially the dendritic cells that phagocytose the influenza virus and help in antigen presentation. We also have T cells, particularly the cytotoxic T lymphocytes, which kill virally infected cells. Finally, we have B cells that enhance the immune response against the virus by producing antibodies as plasma cells. Memory B cells remain after the infection has been cleared and allow for a quicker response against the virus should subsequent infections occur. Long-lived plasma cells also continue to secrete antibodies for months to years after the infection has been cleared. The influenza virus infects only lung epithelial cells. The influenza virus can spread by the inhalation of aerosols containing the virus itself. When someone inhales the virus, it enters the lung and will affect the epithelial cells present. The virus, being enveloped, can fuse with the cell membrane of lung epithelial cells, entering the cells within which they reproduce. New virus leave the infected cell and go on to infect other cells. The infected cell dies when too many viruses bud off from the cell membrane. Immature dendritic cells are professional antigen uptaking cells that constantly sample the environment for antigens. When the dendritic cell detects the influenza virus in tissues, it engulfs it via phagocytosis. Toll-like receptors 7 and 8 recognize single-stranded RNA in endosomes, which is a common feature of viral genomes which are internalized by macrophages and dendritic cells. Toll-like receptors 7 and 8 are intracellular receptors bound to vesicles that are also able to detect single-stranded RNA segments of influenza. The binding of receptors to single-stranded RNA causes cell activation and maturation, the secretion and production of cytokines, the surface expression of CD80 and CD86. Cytokines and CD80 and CD86 are important in the activation of adaptive immune system which ultimately clears the body of infection. At the same time, there is also the loading of influenza virus antigens onto MHC class I molecules. When the virus enters the cell, it is ubiquitinated. The proteasome will digest the virus and release peptides, which enter the endoplasmic reticulum with the help of TAP1 and TAP2 proteins. Within the endoplasmic reticulum, the peptides will be loaded onto MHC class I molecules, forming the PMHC class I molecule. The PMHC class I will bud off from the endoplasmic reticulum and travel towards the Golgi body and to the cell membrane in a secretory vesicle. Eventually, it will be expressed on the cell surface via exocytosis. Mature dendritic cells enter the lymph node via the efferent lymphatic vessels. They have PMHC class I on their surface and they have co-stimulatory molecules CD80 and CD86. They present antigens to naive CD8 T cells. Naive CD8 T cells are activated by the binding of CD80 to CD28 on their surface and by the binding of PMHC class I to T cell receptors. They will differentiate into cytotoxic T lymphocytes. Cytotoxic T lymphocytes will leave the lymph nodes via the efferent lymphatic vessel. They arrive at the site of infection and begin killing infected cells. The cytotoxic T lymphocytes has granules that contains two important enzymes, perforin and gramzyme. When the cytotoxic T lymphocytes T cell receptors binds to an infected cell's PMHC class I molecule, it triggers the release of granule contents. Perforin causes pores to be formed in the cell membrane and gramzyme causes the infected cells to undergo apoptosis. Cytotoxic T lymphocytes can also kill infected cells by triggering apoptosis in infected cells after FASL, a ligand on their surface, binds to the FAST receptors on infected cells. In the lymph node, there is also the activation of mature naive B cells. CD4 T cells are important in the activation of B cells in response to the flu virus. Th2 and Th1 can help in the activation of B cells, and can initiate class switching and infinity maturation. A B cell receptor can bind to antigens found on the influenza virus, and the antigen is endocytosed, processed, and loaded onto the MHC class II molecule to be presented on the surface of the B cell. The PMHC class II T cell receptors bridge, and the binding of CD40 on the B cell to CD40 ligand on the Th2, along with the secretion of interleukins 4 and 5, help in the activation of B cells. Activated B cells proliferate and form a germinal center in the lymph node. There, the B cells become memory B cells and plasma cells after undergoing class switching and somatic hypermutation. In class switching, a B cell will change the isotype of the antibody it produces. IgM, a B cell receptor that is expressed on the surface of all mature naive B cells, can switch to IgA, IgE, or IgG. The binding of CD40 to CD40 ligands initiate class switching from IgM to IgA and IgE. In viral immunity, IgG is of utmost importance. IgG is an antibody that is produced by memory B cells and is secreted into the bloodstream as a monomer. It is very effective at neutralizing the influenza A virus, preventing it from further infecting other cells because it prevents membrane fusion, which is needed for the entry of the virus. The binding of IgG to the virus also opsonizes the virus, which enhances phagocytosis of the virus. IgA also helps in viral immunity. It exists as a dimeric antibody on mucosal surfaces. While the lung has no mucosal, the trachea and the larger bronchi do. There, IgA can neutralize the virus before it can infect other cells. The immune system goes on battling influenza virus until the infection has been cleared. After the infection has been cleared, any infected B or T cells involved will die. However, IgG and IgA are still secreted long after infection with influenza. IgG is secreted by memory B cells while IgA is secreted by long-lived plasma cells. Bone marrow and lung microenvironments foster the maintenance of the long-lived plasma cells.